Death has rules. The heart stops. The brain shuts down. The story ends. But what if I told you that across this planet, death is being rewritten by invisible puppet masters who turn the deceased into mindless servants? What if the zombie apocalypse isn't coming, it's already here? You've seen them stumble through your favourite horror movies. Shambling corpses with rotting flesh, seeking human brains. But here's the terrifying truth that Hollywood missed. Real zombies don't need to die first. They're walking among us right now, their minds hijacked, their bodies controlled by microscopic overlords who've been perfecting the art of possession for hundreds of millions of years. An ant climbs a blade of grass. Seems normal, except this ant has been dead for 3 days. Its muscles are being operated by fungal threads that have woven themselves through every fibre of its body. The ant's brain is still alive, still aware, trapped inside a corpse that no longer obeys its commands. The fungus pilots this biological robot to the perfect height, forces it to bite down with a death grip, and then it waits. A house cat stalks through your neighbourhood. But the mouse it's hunting isn't running away. Instead, it's drawn to the cat's scent like a moth to flame. The mouse's brain has been chemically rewired by a microscopic parasite that needs the cat to complete its life cycle. The mouse thinks it's making its own choices, but every neuron firing in its brain is dancing to the parasite's tune. You might be thinking this is nature's horror show, something that happens to other creatures in other places. But here's what will keep you awake tonight. If you're listening to this, there's a 1 in 3 chance that your brain is already hosting one of these puppet masters. It's been there for months, maybe years, quietly adjusting your personality, influencing your decisions, making you more aggressive or more trusting, depending on its needs. The zombie apocalypse isn't a future threat. It's happening right now in bodies both living and dead, in minds that think they're free but are actually following a script written by creatures so small they're invisible to the naked eye. Welcome to the real world of zombie science, where the boundary between life and death blurs, where free will becomes a question mark, and where the most sophisticated biological technologies on Earth are being deployed by organisms that have no brains, no consciousness, no awareness of their own genius. We're about to journey into a realm where a single-celled parasite can perform neurosurgery with chemical precision, where viruses rewrite the very code of life, and where the survival strategies that have evolved over millions of years make our most advanced robotics look like children's toys. The question isn't whether zombies are real. The question is, who's pulling your strings? And once you know the answer, you'll never look at life, death, or your own decisions the same way again. But to understand how deep this rabbit hole goes, we need to travel to a rainforest where scientists first discovered that death itself can be hacked, where they found something that should have been impossible, a creature that dies yet continues to follow orders with mechanical precision. That creature is an ant, and the force controlling it is about to rewrite everything you thought you knew about the nature of life itself. Chapter 1. The Fungal Strangler. Death Grip in the Canopy. Deep in the rainforests of Thailand, Dr. Charissa de Becker made a discovery that would fundamentally change how we understand death itself. She found an ant frozen in time, its mandibles locked around a leaf in what appeared to be its final moment of life. But this wasn't just any death scene. This was a crime scene, and the evidence was about to reveal a killer so sophisticated that it makes our most advanced technology look primitive. The ant's body position told a story of impossible precision. Not only had it climbed to exactly 25 centimeters above the forest floor, but it had also positioned itself on the north side of the plant stem, where temperature and humidity conditions were optimal for what was about to happen next. The bite force required to penetrate the leaf surface and create that death grip measured 47 times stronger than the ant's normal muscle capacity. The killer responsible for this biological impossibility is a fungus called Ophiocordyceps unilateralis. But calling it just a fungus is like calling a master puppeteer just a person with strings. This organism has evolved over millions of years to perform the most sophisticated act of biological manipulation ever documented in nature. The perfect crime scene. The forensic evidence begins at the cellular level. When researchers examined the ant's tissues under electron microscopy, they discovered something that should have been impossible. The fungal cells had infiltrated every major muscle group in the ant's body. But here's where it gets truly disturbing. The fungus had left the ant's brain almost completely intact. Think about what this means. The ant was aware of what was happening to it. Its brain was processing information, making decisions, trying to send commands to its muscles. But those muscles were no longer under the ant's control. Instead, fungal cells had grown around each muscle fiber, creating what researchers describe as a biological exoskeleton that could override the ant's nervous system. The positioning of the death bite reveals the fungus's understanding of environmental physics that rivals our most sophisticated weather prediction models. The exact height of 25 centimeters above ground level represents the optimal zone where temperature remains stable between 24 and 26 degrees Celsius, while humidity stays consistently between 94 and 96%. These conditions are absolutely critical for the fungus's reproductive cycle. But the fungus doesn't just understand these environmental factors intellectually. It has no brain, no nervous system, no ability to calculate or plan. Yet somehow it navigates the ant's body to this precise location with an accuracy that would make GPS technology jealous. The mechanism behind this navigation involves chemical gradients that the fungus can detect and respond to at the molecular level. The death grip itself represents an engineering marvel that human technology has yet to replicate. The ant's mandibles penetrate the leaf with such force that they actually create microscopic anchors, hooks that lock into the leaf's cellular structure. The fungus then secretes specialized enzymes that harden these connections, creating a biological rivet system that can withstand wind speeds of up to 60 kilometers per hour. Research has revealed that the fungus doesn't just randomly take control of the ant's jaw muscles. It specifically targets the muscles responsible for opening the mandibles while leaving the closing muscles under the ant's control. This creates a biological ratchet system where the ant can bite down but cannot release its grip. The harder the ant tries to escape, the tighter its death grip becomes. Molecular invasion. The invasion begins with a single spore landing on the ant's exoskeleton. This spore is equipped with specialized enzymes that can dissolve the chitin proteins that make up the ant's natural armor. But the fungus doesn't just break down the exoskeleton randomly. It targets specific weak points that correspond to the ant's respiratory system, creating entry points that allow the fungus to bypass the ant's immune defenses. Once inside the ant's body, the fungus begins producing what researchers call invasion filaments. These are specialized structures that can navigate through the ant's tissues with surgical precision. The filaments follow the ant's circulatory system, using the ant's own blood flow to distribute themselves throughout the body. Within 72 hours, the fungus has established colonies in every major organ system. The most sophisticated aspect of this invasion involves the fungus's ability to avoid triggering the ant's immune response. Normal infections cause inflammation, swelling, and other defensive reactions that would interfere with the ant's ability to move and climb. But Ophiocordyceps produces immunosuppressive compounds that essentially convince the ant's immune system that the fungus belongs there. These compounds include molecules that mimic the ant's own cellular recognition signals. The fungus essentially presents forged identification documents to the ant's immune cells, allowing it to operate within the ant's body without detection. This biological stealth technology is so advanced that medical researchers are studying these compounds as potential treatments for autoimmune diseases in humans. The fungus's growth pattern within the ant's body follows a precise timeline that researchers have mapped with microscopic accuracy. Day 1 involves establishing the initial colony sites. Days 2 through 4 focus on infiltrating the nervous system without disrupting essential functions. Days 5 through 7 involve the gradual takeover of motor control systems. By day 8, the ant is no longer controlling its own movements. But the fungus doesn't just take control randomly. It maintains the ant's essential life functions while redirecting its behavior. The ant continues to eat, drink, and perform basic maintenance activities. From the outside, it appears to be functioning normally. Only sophisticated behavioral analysis reveals that the ant is now following a behavioral program written by the fungus rather than its own instincts. Chemical mind control. The chemistry of mind control employed by Ophiocordyceps involves a cocktail of compounds that would make any pharmaceutical company jealous. The primary active ingredient is a molecule called guanobutyric acid, which interferes with the ant's GABA receptors. GABA is the brain's primary inhibitory neurotransmitter, responsible for controlling anxiety, muscle tension, and decision-making processes. By blocking these receptors, the fungus essentially removes the ant's ability to feel fear or hesitation. The ant becomes willing to take risks that would normally trigger powerful survival instincts. This chemical courage is what allows the infected ant to climb to dangerous heights and expose itself to predators without the paralyzing fear that would normally prevent such behavior. But guanobutyric acid is just the beginning. The fungus also produces compounds that affect the ant's dopamine pathways. Dopamine is the neurotransmitter associated with motivation, reward-seeking, and goal-directed behavior. By manipulating these pathways, the fungus can literally reprogram what the ant finds rewarding. Activities that would normally seem pointless or dangerous become irresistibly attractive. The fungus's chemical arsenal includes molecules that affect the ant's circadian rhythms. Normal ants are active during specific hours of the day, but infected ants lose this natural timing. Instead, they become active whenever environmental conditions are optimal for the fungus's reproductive cycle. This represents a complete override of millions of years of evolved behavioral programming. One of the most disturbing aspects of this chemical manipulation involves the fungus's ability to affect the ant's social behavior. Ants are highly social creatures that rely on chemical communication to coordinate colony activities. The fungus produces compounds that disrupt these chemical signals, effectively isolating the infected ant from its colony. The ant becomes a biological island, cut off from the social support systems that normally help ants survive in dangerous environments. The precision of these chemical interventions reveals something profound about the nature of consciousness and free will. The fungus doesn't need to understand what it's doing to the ant's mind. It simply produces specific molecules that have predictable effects on neural circuits. This suggests that consciousness itself might be more chemically determined than we typically assume. Research has identified over 40 different compounds produced by the fungus during the infection process. Each compound serves a specific function in the manipulation process. Some affect motor control, others influence decision-making, and still others modify the ant's sensory perception. Together, these compounds create a comprehensive chemical rewrite of the ant's behavioral programming. Ecological domino effect. The death of a single ant might seem insignificant, but the ecological consequences of fungal zombification ripple through entire forest ecosystems in ways that researchers are only beginning to understand. Each infected ant represents a walking biological weapon that can potentially spread the fungus to dozens of other ants before completing its final programmed behavior. The fungus's reproductive strategy involves creating what researchers call spore bombs from the ant's corpse. After the ant locks into its death grip, the fungus begins converting the ant's internal organs into spore-producing factories. Within 48 hours, the ant's body cavity is filled with millions of microscopic spores, each one capable of infecting a new host. But the fungus doesn't just randomly scatter these spores. It grows specialized structures called sporocarps that emerge from the ant's head and body. These structures are essentially biological artillery pieces that can launch spores with incredible precision. The fungus can actually aim these spores toward areas where healthy ants are likely to travel, maximizing the infection rate. The impact on ant colonies can be devastating. A single infected ant can potentially eliminate an entire colony of several thousand individuals. This has profound effects on the forest ecosystem, since these ants play crucial roles in seed dispersal, soil aeration, and pest control. When ant populations collapse, the entire forest food web begins to shift in unpredictable ways. Some plant species have evolved to depend on specific ant species for their reproductive success. These plants produce seeds with specialized structures that attract ants, which then carry the seeds to new locations. When the fungus eliminates these ant species, the plants lose their primary method of reproduction, leading to population crashes that can affect entire forest canopies. The fungus's impact extends beyond just ants and plants. Many bird species rely on ant colonies as food sources, particularly during breeding season when protein requirements are highest. When ant populations crash, bird reproductive success plummets, leading to cascading effects throughout the forest food web. Researchers have documented cases where fungal outbreaks have altered the competitive balance between different ant species. Some species are more susceptible to infection than others, leading to dramatic shifts in which species dominate particular forest areas. These changes can persist for decades, fundamentally altering the character of forest ecosystems. The fungus's influence on forest dynamics also extends to the decomposition process. Ants play crucial roles in breaking down dead plant material and recycling nutrients back into the soil. When ant populations are reduced by fungal infections, decomposition rates slow dramatically, leading to changes in soil chemistry that affect plant growth patterns. Perhaps most disturbing is the discovery that the fungus can actually modify the forest environment to make it more suitable for its own reproduction. Infected ants tend to select death locations that create microclimates with optimal temperature and humidity conditions. Over time, these death gardens become hotspots for fungal growth that can persist for years, creating permanent infection zones within the forest. The Ophiocordyceps fungus represents just the beginning of our journey into the world of biological puppet masters. Its ability to hijack an ant's body and mind with such precision reveals capabilities that challenge our fundamental assumptions about consciousness, free will, and the nature of life itself. But here's what should truly disturb you. This fungus, sophisticated as it is actually being controlled by something even more mysterious. Deep within the fungal genome, researchers have discovered genetic sequences that don't belong to the fungus. These sequences appear to be viral in origin, suggesting that the fungus itself is being manipulated by an even smaller puppet master. The implications are staggering. If viruses can control fungi, and fungi can control complex animals, what does this mean for the hierarchy of life on Earth? Are we all just biological machines being operated by microscopic overlords that we can't even see? The answer to that question lies in our next investigation, where we'll explore a pathogen that doesn't just control behavior, but actively rewrites the brain's emotional circuitry to create something that looks disturbingly similar to the rage-filled zombies of popular fiction. This pathogen has been with humanity for thousands of years, and its influence on human history is far more profound than you might imagine. Chapter 2. Rage Virus. The Bite That Lives in You. 4,000 years ago, ancient Mesopotamian tablets described something that terrified our ancestors more than war, more than famine, more than death itself. They called it the Hand of the Ghost, a curse that turned beloved family members into snarling, violent strangers who feared water like fire and attacked with mindless fury. These weren't stories of supernatural possession. They were the first recorded cases of humanity's encounter with the most sophisticated neurological hijacker ever discovered, the rabies virus. Every zombie movie you've ever watched owes its existence to this microscopic puppet master. The shambling gait, the mindless aggression, the unstoppable drive to bite and spread infection. These aren't Hollywood inventions. They're precise biological programs written by a virus that has been perfecting the art of behavioral control for millions of years. Ancient fear, the demon that lived in saliva. The ancient world knew this enemy well, though they couldn't see it. Egyptian papyri from 2300 BCE described rabid dogs as possessed by demons. Greek physicians called it lyssa, meaning frenzy or madness. But the most chilling account comes from ancient India, where Sanskrit texts describe victims who bark like dogs, fear their own shadows, and see water as liquid fire. These weren't superstitious explanations. They were accurate clinical observations of what happens when a virus rewrites the operating system of a mammalian brain. The ancient physicians couldn't understand the mechanism, but they recognized the pattern. A bite, followed by a strange wound that wouldn't heal normally, then weeks of apparent recovery, and finally the transformation into something that was no longer quite human. What our ancestors witnessed was the world's first documented case of biological warfare. A pathogen that doesn't just kill its host, but reprograms them into a transmission machine. The rabies virus had already spent millions of years perfecting this strategy, turning infected animals into living weapons designed for a single purpose: to bite and spread. The ancient fear wasn't irrational. It was the recognition of something that challenged the very foundations of human identity. If a simple bite could transform a loving mother into a creature that would attack her own children, what did that say about the nature of consciousness, of free will, of the stability of human personality? Neuroinvasion highway, the virus that travels by nerve. Understanding how rabies transforms behavior requires following its journey through the body, and this journey reads like a biological thriller. The virus doesn't spread through the bloodstream like most pathogens. Instead, it uses something far more insidious: your own nervous system as a highway to the brain. When rabies virus particles enter through a bite wound, they don't immediately cause infection. Instead, they seek out peripheral nerve endings, the microscopic cables that carry signals between your brain and muscles. The virus attaches to acetylcholine receptors, molecular docking stations that normally receive signals from motor neurons. Here's where the viral genius becomes apparent. Once attached, the virus hijacks the nerve's own transportation system. Neurons naturally move materials back and forth along their length using molecular motors called dynein and kinesin. Rabies virus commandeers these motors, forcing them to carry viral particles along the nerve fiber toward the central nervous system. The virus travels at a rate of about 12 to 100 millimeters per day, depending on the nerve pathway. This explains why rabies has such a variable incubation period. A bite on the hand might take months to reach the brain, while a bite on the face could cause symptoms in days. The virus is literally crawling through your nervous system, using your own neural infrastructure as its personal highway. But the most disturbing aspect of this journey is how the virus modifies the nerves as it travels. It doesn't just use the highway, it rebuilds it. The virus alters the expression of genes in infected neurons, changing how they function even before it reaches the brain. By the time the virus arrives at the central nervous system, it has already begun rewiring the very pathways it will use to control behavior. This isn't random viral replication. This is targeted strategic modification of the host's neural networks, preparing the groundwork for the behavioral changes that will follow. The virus is literally paving the road for its own success. Hydrophobia horror, the fear that serves the virus. The most iconic symptom of rabies is hydrophobia, the pathological fear of water that causes infected individuals to recoil in terror from any liquid. This isn't just a cruel side effect of viral infection. It's a precisely engineered biological mechanism that maximizes viral transmission, and understanding how it works reveals the terrifying sophistication of viral behavioral control. The rabies virus targets specific regions of the brainstem responsible for swallowing and respiratory control. As the virus replicates in these areas, it causes inflammation that makes swallowing progressively more difficult and painful. But the virus doesn't stop there. It also triggers hypersensitivity in the neurons that control the larynx and pharynx, making any attempt to swallow feel like swallowing broken glass. The psychological component is equally calculated. The virus affects the amygdala and associated fear processing centers, creating an intense, irrational terror of water and other liquids. The mere sight of water can trigger violent muscle spasms and panic attacks. Even the sound of running water can cause infected individuals to flee in terror. Why would a virus evolve such a seemingly counterproductive mechanism? The answer lies in viral transmission strategy. When an infected animal or person cannot swallow, saliva accumulates in the mouth and throat. This saliva contains millions of viral particles, perfectly positioned for transmission through the next bite. The hydrophobia ensures that the viral load remains concentrated in the oral cavity, ready for spread. The virus has essentially turned the host's mouth into a biological weapon loaded with infectious material. The inability to swallow isn't a bug in the system, it's a feature. The virus has engineered its host into a more efficient transmission vector by preventing the dilution of viral particles through normal swallowing reflexes. Even more disturbing is how the virus maintains this state. As the infection progresses, the host experiences intense thirst but cannot drink. This creates a psychological torture that drives increasingly desperate behavior. The combination of mounting thirst, fear of water, and accumulated saliva creates a perfect storm of viral transmission potential. The virus has essentially engineered a biological catch-22. The very thing the host desperately needs, water, becomes the thing they fear most. This isn't accidental. This is millions of years of evolutionary refinement, creating a system so efficient at viral spread that it's become the blueprint for fictional zombie viruses in popular culture. The ultimate betrayal. Viruses forcing mothers to attack their young. The final stage of rabies infection reveals the most disturbing aspect of viral behavioral control, the complete inversion of natural instincts. The virus doesn't just make hosts aggressive, it specifically targets and overrides the deepest evolutionary programming in the mammalian brain, including the maternal instinct to protect offspring. Female mammals infected with rabies will attack their own young with the same fury they show towards strangers. Maternal cats will bite their kittens, infected dogs will snap at their puppies, and human mothers in the final stages of rabies have been documented attacking their own children. This isn't random violence. It's the virus systematically dismantling the host's most fundamental behavioral programming. The neurological mechanism behind this betrayal involves the virus's assault on the limbic system, particularly the areas responsible for social bonding and parental behavior. The virus specifically targets neurons that produce oxytocin and vasopressin, the hormones responsible for bonding and protective instincts. As these neurons become infected and inflamed, the chemical signals that normally promote nurturing behavior are disrupted. Simultaneously, the virus amplifies activity in the amygdala and hypothalamus, regions associated with fear and aggression. The result is a complete inversion of normal emotional responses. The faces that once triggered protective instincts now trigger attack responses. The cries that once prompted care now prompt aggression. This behavioral modification serves the virus's transmission strategy perfectly. Infected mothers don't just fail to protect their young, they actively become vectors for spreading the virus to the next generation. The virus has turned the strongest bond in nature into a transmission pathway. The viral programming is so complete that infected animals show no recognition of their offspring. Brain scans of rabies-infected animals reveal that the neural pathways responsible for recognizing familiar individuals become functionally disconnected. The virus has essentially erased the host's ability to distinguish between family members and strangers. But perhaps most disturbing is how this occurs while much of the host's cognitive function remains intact. Unlike other forms of brain damage that cause global impairment, rabies creates very specific behavioral changes while leaving other mental faculties relatively preserved. The host can still recognize objects, solve problems, and process information, but they can no longer access the emotional and social programming that makes them who they are. The virus has essentially performed a targeted deletion of the host's social operating system while leaving the rest of their mental hardware intact. This creates the uncanny valley effect that makes rabies so terrifying. The infected individual is still recognizably themselves, but the essential qualities that made them human or made them a caring parent have been surgically removed by viral action. Rabies represents the perfect fusion of lethality and behavioral control. The virus doesn't just kill, it transforms its host into a living transmission machine programmed with a single directive: bite and spread. Every symptom, from the initial wound sensitivity to the final stages of hydrophobic fury, serves the virus's reproductive strategy. The rabies virus has been called nature's perfect biological weapon, and for good reason. It spreads through direct contact, has an extremely high fatality rate, and causes behavioral changes that maximize transmission. It's the closest thing to a real zombie virus that exists in nature, and it's been quietly perfecting its craft for millions of years. But rabies operates on a timeline measured in days and weeks. The virus kills its host relatively quickly, limiting its transmission potential. What if there were pathogens that could control behavior not for days, but for months or even years? What if there were parasites that could influence their host's behavior while keeping them alive and functional, spreading their influence through entire populations without anyone noticing? The rabies virus showed us that minds can be hijacked and behavior can be controlled. But it was just the beginning. Deep in the natural world there are parasites that have mastered the art of long-term behavioral manipulation, turning their hosts into unwitting accomplices in elaborate multi-year schemes that would make the most sophisticated spy thriller look like a children's story. These parasites don't just control their hosts. They engineer their entire life stories, orchestrating elaborate sequences of behavior that can span multiple species and several years. They've turned biological manipulation into an art form, and their masterpieces are walking among us right now, following scripts they don't even know they're reading. Chapter 3. Parasite Puppeteers. Long Game of the Undead. The virus that hijacks your brain in days seems like nature's ultimate weapon. But there's something far more sinister lurking in the biological world. Something that doesn't need to kill you quickly to get what it wants. These creatures play the long game, and they've turned patience into the most devastating weapon in the natural world. Meet the puppet masters who don't just steal your body for a few violent days. They steal your entire life, rewriting your deepest instincts, your most basic drives, your will to live itself. And they do it so subtly, so completely that you'll walk willingly to your own execution, convinced that every step is your own choice. The worm's cunning. The lancet liver fluke, Dicrocoelium dendriticum, has solved one of biology's greatest puzzles: how to get from the inside of an ant to the inside of a sheep. The journey requires precision timing, behavioral manipulation, and a level of biological engineering that would make our smartest roboticists weep with envy. Inside the ant's body, thousands of juvenile flukes are developing, but they face an impossible problem. Their life cycle demands they reach the stomach of a grazing mammal, yet they are trapped inside a creature that spends its entire life avoiding those very predators. The solution the fluke has evolved is nothing short of biological genius. As night falls, something extraordinary happens inside the ant's brain. A single fluke, different from all the others, begins its migration. This specialist fluke doesn't develop reproductive organs like its siblings. Instead, it sacrifices its own future for a greater purpose. It crawls directly into the ant's brain and positions itself near the subesophageal ganglion, the cluster of neurons that controls the ant's basic motor functions. The ant wakes up the next morning, but it's no longer the same creature. Its normal behavioral patterns have been overwritten by microscopic chemical signals from the fluke in its brain. Instead of returning to the colony with the other workers, this ant begins climbing up grass stems, up plant stalks, driven by an inexplicable compulsion to reach the very tips of vegetation where grazing animals feed. Here's where the fluke's strategy reveals its true sophistication. The ant doesn't just climb randomly. The parasite has hijacked the ant's circadian rhythm, its internal biological clock. The ant climbs only during the cool morning and evening hours when sheep and cattle are most likely to graze. During the heat of midday, when grazing animals seek shade, the ant returns to normal behavior, hiding in the grass to avoid deadly dehydration. This cycle continues for days, sometimes weeks. The ant becomes a living alarm clock, programmed to place itself in mortal danger precisely when the fluke needs it most. The moment a sheep's mouth closes around that blade of grass, the fluke's patient strategy pays off. The ant dies, but the thousands of juvenile flukes in its body have reached their destination, the sheep's digestive system, where they'll mature and begin the cycle anew. Sacrificial fish. In the salt marshes of California, another puppet master has perfected an even more audacious strategy. The trematode Euhoplorchis californiensis faces a similar challenge. It needs to get from fish to bird, but fish naturally avoid the surface waters where birds hunt. The solution this parasite has evolved defies everything we thought we knew about biological camouflage. The killifish, Fungulus parvipinis, is a master of survival in these harsh tidal environments. Its silver sides and dark back provide perfect camouflage against both aerial and underwater predators. These fish have survived millions of years by being virtually invisible. But when infected with Euhoplorchis, they transform into living disco balls. The parasite doesn't just change the fish's behavior, it fundamentally alters its appearance. Infected fish develop brilliant white and silver patches that flash and shimmer as they swim. These aren't random color changes. The parasite has hijacked the fish's chromatophore cells, the specialized cells that control coloration, turning them into biological spotlights. But the visual display is only part of the strategy. The parasite also rewrites the fish's neural programming, driving it to swim in rapid, erratic patterns near the water's surface. The combination of flashing colors and spastic movements creates what researchers call a supernormal stimulus, a signal so exaggerated that it triggers an irresistible feeding response in birds. Think about what this means. The parasite has simultaneously solved multiple complex problems: how to make a camouflaged animal conspicuous, how to overcome millions of years of predator avoidance instincts, and how to time these behavioral changes to coincide with bird feeding patterns. It's biological engineering at a level that makes our most advanced biotechnology look primitive. The timing of this transformation is crucial. The fish only develop these conspicuous displays once the parasites have reached the right stage of development. Too early, and the parasites die with the fish. Too late, and they miss their window of opportunity. The parasites have evolved internal chemical clocks that synchronize their manipulation with their own life cycle requirements. Zombie snails. The green-banded broodsac, Leucochloridium paradoxum, has achieved what might be the most visually stunning feat of biological puppeteering on Earth. This parasite doesn't just change its host behavior, it transforms the host body into a living lure so convincing that birds mistake it for their favorite food. The amber snail begins its day like any other, sliding along the forest floor, feeding on decaying leaves. But inside its body, something extraordinary is happening. The parasite has been growing, developing, preparing for a performance that will determine whether its genetic lineage continues or ends forever. As the parasite matures, it begins to grow into the snail's tentacles, the eye stalks that the snail uses to navigate its world. But instead of simply occupying space, the parasite transforms these organs into biological masterpieces of deception. The infected tentacles swell to many times their normal size, developing thick bands of bright green and brown that pulse rhythmically, creating an almost hypnotic display. The pulsing isn't random. The parasite has evolved to mimic the exact coloration and movement patterns of caterpillars, the preferred food of many bird species. The frequency of the pulsing, the intensity of the colors, even the way the tentacles move as the snail crawls, every aspect has been fine-tuned by natural selection to create the most convincing caterpillar impersonation possible. But here's where the strategy becomes truly diabolical. The parasite doesn't just make the snail look like a caterpillar, it forces the snail to behave like one. Infected snails abandon their normal preference for dark, hidden places and instead seek out brightly lit areas where they're most visible to birds. They climb higher than normal, exposing their transformed tentacles to maximum effect. The snail's brain is still functioning, still processing the world around it, but its most basic survival instincts have been overwritten. It's as if the parasite has installed new software that runs on the snail's existing hardware, completely changing the output while leaving the underlying system intact. When a bird finally takes the bait and attacks the snail, it typically goes for the conspicuous tentacles, exactly what the parasite wants. The bird gets what it thinks is a caterpillar. The snail often survives with regenerated tentacles, and the parasite reaches its destination in the bird's digestive system. It's a strategy so elegant, so perfectly calibrated, that it seems almost too sophisticated to have evolved naturally. Evolutionary arms race. But evolution never stops, and neither do the hosts. For every parasitic strategy that evolves, natural selection drives the development of countermeasures. What we're witnessing is biological warfare at its most fundamental level, played out over millions of years between species locked in an eternal struggle for survival. The ants infected by liver flukes should be evolutionary dead ends. They climb to exposed positions, making themselves vulnerable to predators, and they do it repeatedly until they die. Any genetic variants that made ants more resistant to this manipulation should spread rapidly through the population. Yet the parasites persist, and they're winning. The reason becomes clear when we examine the molecular battleground inside the ant's brain. The fluke produces a cocktail of neurochemicals that includes modified versions of the ant's own neurotransmitters. It's not just hijacking the ant's neural networks, it's speaking the ant's own chemical language, but with a foreign accent. Some ant populations have evolved partial resistance. These ants carry genetic variations that make their neurons less responsive to the fluke's chemical signals. But this resistance comes at a cost. The same neural modifications that protect against parasites also reduce the ant's ability to respond to legitimate chemical signals from its own colony. The parasites, meanwhile, are evolving countermeasures. New strains of liver flukes produce more potent versions of their neural cocktails, or they target different neural pathways entirely. It's an arms race where both sides are constantly innovating, constantly adapting, constantly trying to stay one step ahead. This isn't just about ants and flukes. Similar battles are playing out in ecosystems around the world. The killifish are evolving better camouflage and stronger predator avoidance instincts. The trematodes are developing more sophisticated ways to override these defenses. The snails are improving their ability to detect and resist parasitic infections. The brood sacs are finding new ways to manipulate their hosts. What emerges from this biological arms race is a level of complexity that defies our traditional understanding of evolution. These aren't simple predator-prey relationships. They're intricate dances of deception and resistance, manipulation and counter-manipulation, played out at the molecular level with stakes that couldn't be higher. The parasites we've seen so far are master manipulators, but they're still playing by certain rules. They need their hosts to survive long enough to complete their life cycles. They need to balance manipulation with preservation. But there's another type of parasite that has thrown these rules aside entirely, one that has discovered something far more insidious than behavioral control. This parasite doesn't just change what you do, it changes who you are. And the most terrifying part? It might already be living inside your skull, quietly rewriting your personality while you sleep. The question isn't whether it's there. The question is, how long has it been watching you through your own eyes? Chapter 4. The Ghost in Your Brain. Toxoplasma's Human Gambit. Every parasite we've encountered so far operates with brutal efficiency. The fungus grows through ant muscles like organic puppet strings. The rabies virus hijacks neural pathways with chemical sledgehammers. But what if I told you there's a parasite so sophisticated, so precise in its manipulation, that it can alter human personality without you ever knowing it's there? This microscopic puppet master doesn't need to kill its host to control them. It doesn't need to cause obvious symptoms or dramatic behavioral changes. Instead, it works in the shadows of your mind, making subtle adjustments to your brain chemistry, your decision-making processes, your very sense of self. And the most terrifying part? It's already inside nearly 2 billion people on this planet. The parasite is called Toxoplasma gondii, and it's about to reveal just how thin the line really is between your thoughts and someone else's agenda. Cat Mafia Scheme, the rodent mind hack. To understand how Toxoplasma controls human behavior, we first need to witness its masterpiece of neural manipulation in action. Deep in the brain of a laboratory mouse, a microscopic drama is unfolding that will rewrite everything you thought you knew about fear, attraction, and free will. This mouse should be terrified. Every instinct evolution has given it over millions of years screams that the scent it's detecting means certain death. Cat urine, the chemical signature of its most dangerous predator. But instead of fleeing, this mouse is doing something that should be impossible. It's moving toward the smell, not cautiously, not reluctantly, but with what can only be described as eager curiosity. The mouse's olfactory neurons are firing normally. Its brain is processing the cat scent correctly. Every other fear response in its body remains intact. Show it a snake and it runs. Play a loud noise and it startles. But present it with cat odor and something extraordinary happens. The fear circuits shut down and attraction circuits light up like a Christmas tree. This is Toxoplasma gondii at work, and what it's doing to this mouse's brain represents one of the most sophisticated examples of neurochemical manipulation in the natural world. The parasite has spent its entire evolutionary history perfecting a single goal, getting from inside a mouse to inside a cat. And it's discovered that the fastest way to achieve this is to hack the mouse's most fundamental survival instinct. But here's where the story becomes truly unsettling. Toxoplasma doesn't just suppress fear randomly. It doesn't make mice unafraid of everything. Instead, it performs what can only be described as neurochemical surgery, targeting specific neural pathways while leaving others completely intact. The infected mouse remains afraid of open spaces, afraid of bright lights, afraid of loud noises. But when it comes to cats, the parasite has literally rewired its brain to transform terror into attraction. Scientists have traced exactly how this manipulation works at the molecular level. Toxoplasma secretes proteins that interfere with the mouse's production of dopamine, the neurotransmitter associated with pleasure and reward. But it doesn't flood the brain with dopamine indiscriminately. Instead, it increases dopamine production specifically in brain regions that process cat odors. The result is that the mouse experiences the scent of its predator as genuinely pleasurable, like a drug addict getting a hit. This isn't just behavior modification. This is personality reconstruction. The parasite has taken a creature whose entire evolutionary history has been shaped by avoiding cats, and it's turned that creature into something that actively seeks out cats. It's the biological equivalent of reprogramming a computer's operating system, except the computer is a living brain, and the programmer is a single-celled organism with no brain of its own. The mouse thinks it's making its own choices. From its perspective, it's simply following its instincts, pursuing something that feels good, exploring something that seems interesting. It has no way of knowing that those instincts have been hijacked, that its sense of what feels good has been chemically altered, that its curiosity is being guided by a microscopic puppet master with its own agenda. And this is where the story takes a disturbing turn toward humans. Because Toxoplasma gondii doesn't just infect mice. It infects any warm-blooded animal it can reach, including the estimated 2.5 billion people currently carrying this parasite in their brains. The question that keeps researchers awake at night is simple. If Toxoplasma can rewrite a mouse's most basic survival instincts, what might it be doing to ours? Silent human invasion, the personality hijack. The numbers alone should terrify you. 30% of the human population. That's roughly 2.5 billion people walking around with a brain parasite that we know can fundamentally alter behavior in other mammals. But here's what makes this statistic truly unsettling. Until recently, we assumed these human infections were completely harmless. We were wrong. The first clue came from traffic accidents. Researchers in the Czech Republic noticed something odd in their data. People who had been in car crashes were significantly more likely to test positive for Toxoplasma infection. At first, this seemed like a statistical fluke. Then the pattern appeared in other countries. Then researchers started looking at other types of risky behavior, and the correlations became impossible to ignore. People infected with Toxoplasma are more likely to start their own businesses. They're more likely to engage in extreme sports. They're more likely to have multiple sexual partners. They're more likely to be involved in motorcycle accidents. They're more likely to attempt suicide. The parasite that makes mice fearless around cats appears to be making humans fearless around danger. But the manipulation goes deeper than just risk-taking. Toxoplasma infection correlates with increased rates of schizophrenia, bipolar disorder, and aggressive behavior. It correlates with changes in personality traits that psychologists can measure with scientific precision. Infected individuals score higher on measures of suspicion, jealousy, and dogmatism. They're more likely to disregard social rules and less likely to trust authority figures. The correlation with schizophrenia is particularly striking. People with schizophrenia are 2 to 3 times more likely to be infected with Toxoplasma than the general population. The parasite has been found in higher concentrations in the brains of people who died by suicide. Countries with higher rates of Toxoplasma infection show higher rates of neuroticism and anxiety disorders at the population level. These aren't just correlations anymore. Researchers have identified the specific biological mechanisms by which Toxoplasma alters human brain function. The parasite forms cysts in brain tissue, particularly in areas associated with emotion, decision-making, and personality. These cysts can persist for decades, continuously releasing proteins that interfere with normal neurotransmitter function. Just as it does in mice, Toxoplasma alters dopamine production in humans, but it also affects other neurotransmitter systems, including serotonin and GABA, the brain's primary inhibitory neurotransmitter. The result is a complex pattern of neurochemical changes that can subtly influence everything from mood to decision-making to social behavior. The parasite's effects on human personality aren't random. They follow predictable patterns that seem designed to increase the likelihood of transmission. Infected individuals are more likely to engage in behaviors that could expose them to cat feces, the parasite's primary source of new hosts. They're more likely to own cats, more likely to clean litter boxes, more likely to engage in gardening where cat feces might be present. But the manipulation extends beyond direct transmission opportunities. Toxoplasma appears to alter human social behavior in ways that could increase the parasite's spread through the population. Infected individuals are more extroverted, more likely to seek out social situations, more likely to engage in behaviors that could facilitate the spread of the parasite to new hosts. The gender differences are particularly revealing. Men and women respond differently to Toxoplasma infection, suggesting that the parasite has evolved different strategies for manipulating different types of hosts. Infected men tend to become more aggressive, more suspicious, more likely to break social rules. Infected women tend to become more trusting, more socially outgoing, more likely to engage in risky sexual behavior. These aren't conscious choices. The people experiencing these personality changes have no idea they're being manipulated. They experience their altered behavior as genuine expressions of their true selves, as natural responses to their environment, as rational decisions based on their own preferences and values. The implications are staggering. If Toxoplasma can influence human personality, decision-making, and social behavior, then what we consider to be individual free will might be partially under the control of a microscopic parasite. The thoughts you think are your own, the decisions you make, the risks you take, the people you trust, the way you interact with the world, all of these might be influenced by a single-celled organism that has its own evolutionary agenda. Dopamine heist, the neurochemical crime. Understanding how Toxoplasma manipulates human behavior requires diving into the most sophisticated biological crime scene ever discovered. This isn't a smash-and-grab operation. This is a precision heist targeting the very chemicals that make us who we are, and the evidence is written in the language of molecules. The crime begins the moment Toxoplasma enters a human brain cell. Most parasites are satisfied with finding a safe place to hide and reproduce. But Toxoplasma has evolved a far more ambitious agenda. It doesn't just want to survive inside your brain. It wants to modify your brain to serve its purposes, and it accomplishes this through one of the most elegant molecular manipulations in the biological world. The parasite's primary target is dopamine, the neurotransmitter that drives motivation, reward-seeking, and pleasure. But Toxoplasma doesn't just increase dopamine production randomly. Instead, it has evolved the ability to manufacture its own dopamine-producing enzymes, essentially turning infected brain cells into drug factories under the parasite's control. The key enzyme is called tyrosine hydroxylase, and it's the rate-limiting step in dopamine production. Normally, human brain cells produce this enzyme in carefully regulated amounts, maintaining precise control over dopamine levels. But Toxoplasma has evolved to produce its own version of this enzyme, flooding infected brain regions with dopamine at levels that would be impossible through normal human physiology. The result is a neurochemical hijacking of unprecedented sophistication. The parasite doesn't just alter existing dopamine pathways, it creates new ones. Brain scans of infected individuals show increased dopamine activity in regions where the parasite has formed cysts, particularly in areas associated with risk assessment, fear processing, and reward evaluation. But dopamine is just the beginning. Toxoplasma also interferes with GABA, the brain's primary inhibitory neurotransmitter. GABA is what keeps us from acting on every impulse, what makes us pause before taking risks, what provides the mental brakes that prevent reckless behavior. By disrupting GABA signaling, the parasite effectively removes these mental inhibitions, making infected individuals more likely to act on impulses they would normally suppress. The parasite's manipulation of serotonin is equally sophisticated. Serotonin influences mood, social behavior, and aggression. Toxoplasma has been found to alter serotonin production in ways that increase social approach behavior while simultaneously increasing aggression and dominance seeking. It's as if the parasite has found a way to make people more social and more aggressive at the same time. Perhaps most remarkably, Toxoplasma appears to manipulate the brain's immune system as well. The parasite secretes proteins that interfere with microglial cells, the brain's resident immune cells. This isn't just about avoiding detection. The parasite actually hijacks these immune cells, turning them into unwitting accomplices in its neurochemical manipulation. Infected microglial cells release inflammatory cytokines that affect neurotransmitter production throughout the brain. These cytokines can influence everything from mood to sleep patterns to cognitive function. The parasite has essentially co-opted the brain's own immune system to carry out its neurochemical agenda. The precision of this manipulation is breathtaking. Toxoplasma has evolved specific proteins that target specific neurotransmitter receptors. It has developed enzymes that can synthesize neurotransmitters more efficiently than human cells. It has created molecular mechanisms that can alter gene expression in infected neurons, changing not just how these cells function, but what proteins they produce. Laboratory studies have confirmed these mechanisms with shocking precision. Researchers can now trace the molecular pathway from Toxoplasma infection to behavioral change. They can measure the increase in dopamine production, quantify the changes in GABA signaling, and correlate these neurochemical alterations with specific behavioral modifications. The evidence is overwhelming. Toxoplasma doesn't just hide in the brain. It actively remodels the brain's chemistry to serve its own purposes. It's not just a passenger. It's a driver. And it's been driving human behavior for thousands of years without us even knowing it was behind the wheel. The implications for human consciousness and free will are profound. If a microscopic parasite can alter the fundamental chemistry of human thought and decision-making, then what we consider to be our authentic selves might be partially under the control of organisms with their own evolutionary agendas. The gender divide, rewiring masculinity and femininity. The most disturbing aspect of Toxoplasma's manipulation might be how it exploits the fundamental differences between male and female brains. This parasite hasn't just evolved to control human behavior, it's evolved different strategies for controlling men and women, as if it understands something about human psychology that we're only beginning to discover ourselves. The pattern emerged from decades of research across multiple countries and cultures. When scientists examined the personality changes associated with Toxoplasma infection, they found something unexpected. The parasite doesn't affect everyone the same way. Instead, it appears to push men and women in opposite directions, amplifying certain traits while suppressing others. Infected men become more aggressive, more suspicious, more likely to break social rules and disregard authority. They score higher on measures of jealousy and dominance-seeking. They're more likely to engage in risky behaviors, more likely to start fights, more likely to pursue multiple sexual partners. The parasite seems to be turning up the volume on traditionally masculine traits while dialing down impulse control and social conformity. But infected women show a completely different pattern. They become more trusting, more socially outgoing, more likely to seek out new social connections. They score higher on measures of warmth and social approach behavior. They're more likely to be sexually promiscuous, but in a way that seems driven by social connection rather than dominance or aggression. These aren't subtle differences. The personality changes are measurable using standard psychological assessments and appear consistently across different populations. It's as if the parasite has evolved to exploit the neurochemical differences between male and female brains to achieve different behavioral outcomes. The biological mechanisms underlying these gender differences are becoming clear. Male and female brains respond differently to dopamine, the neurotransmitter that Toxoplasma manipulates most directly. In men, increased dopamine tends to enhance aggression, risk-taking, and dominance-seeking behaviors. In women, increased dopamine more often enhances social approach behaviors and trust. But Toxoplasma's manipulation goes beyond dopamine. The parasite affects testosterone and estrogen signaling in ways that amplify these gender differences. Infected men show increased testosterone levels, which correlates with increased aggression and risk-taking. Infected women show changes in estrogen sensitivity that correlate with increased social approach behavior and trust. The evolutionary logic becomes clear when you consider the parasite's ultimate goal: transmission to new hosts. For a parasite that spreads through cat feces, having infected humans engage in behaviors that increase contact with cats or with other infected individuals serves the parasite's interests. But men and women might serve these interests in different ways. More aggressive, risk-taking men might be more likely to engage in behaviors that expose them to cat feces, such as outdoor activities, gardening, or handling cats. More social, trusting women might be more likely to share close contact with other infected individuals, potentially facilitating sexual transmission or creating social networks that increase the parasite's spread. The sexual behavioral changes are particularly striking. Both infected men and women are more likely to have multiple sexual partners, but for different reasons. Men appear to be driven by increased dominance and aggression, while women appear to be driven by increased social approach and trust. Both patterns could serve the parasite's interests by increasing the likelihood of sexual transmission. But the manipulation extends beyond sexual behavior. Infected men are more likely to engage in entrepreneurial activities, starting businesses and taking financial risks. Infected women are more likely to engage in caregiving behaviors, working in professions that involve close contact with other people. Both patterns could serve the parasite's interests by creating different types of social networks that facilitate transmission. The implications are deeply unsettling. If Toxoplasma can manipulate gender-specific behaviors with such precision, it suggests that the parasite has evolved an understanding of human psychology that rivals our own scientific knowledge. It's as if the parasite has been conducting its own behavioral psychology experiments for millions of years, evolving increasingly sophisticated strategies for manipulating human behavior. These findings challenge our understanding of gender differences themselves. How much of what we consider to be naturally masculine or feminine behavior might be influenced by the presence of microscopic parasites? How much of human personality, human social behavior, human sexuality might be under the partial control of organisms with their own evolutionary agendas? The answers to these questions are still emerging, but the evidence is clear. Toxoplasma gondii has evolved the ability to manipulate human behavior in gender-specific ways, pushing men and women in directions that serve the parasite's interests, while making these behavioral changes feel natural and voluntary to the infected individuals. Standing at the edge of this neurochemical rabbit hole, we face a truth that challenges everything we thought we knew about human nature. Toxoplasma gondii has revealed that the boundary between self and other, between our thoughts and someone else's agenda, is far more permeable than we ever imagined. This microscopic puppet master has been quietly influencing human behavior for millennia, shaping personalities, driving decisions, and altering the very fabric of human society. It's been conducting the largest behavioral modification experiment in human history, and we're only just beginning to understand the scope of its influence. But Toxoplasma's subtle manipulation pales in comparison to what happens when microscopic puppet masters stop working in shadows and start causing global chaos. Because sometimes parasites don't just change individual behavior. Sometimes they change the behavior of entire populations, creating mass behavioral modifications that can reshape civilization itself. The question that should terrify you isn't whether you're infected with Toxoplasma. The question is, what happens when a behavioral parasite stops targeting individuals and starts targeting entire societies? What happens when the puppet master decides that subtle influence isn't enough and it's time to pull the strings of humanity itself? The answer lies in the darkest chapters of human history, where mass behavioral changes swept across populations like invisible plagues, where entire civilizations found themselves dancing to the rhythm of microscopic drums they couldn't hear. And once you understand how these biological puppeteers can orchestrate mass behavior modification, you'll never look at human history, human society, or human nature the same way again. Chapter 5, Apocalypse Now, When Parasites Go Global. Individual mind control is terrifying enough. One ant, one mouse, one human brain hijacked by microscopic invaders. But what happens when these puppet masters don't just target individuals? What happens when they orchestrate mass behavioral changes across entire populations, reshaping the very fabric of society itself? The year was 1918. The Great War was ending, but a new enemy was sweeping across the globe. Within months, it would kill more people than 4 years of brutal warfare. But this pathogen didn't just kill. It did something far more unsettling. It changed how people behaved on a planetary scale. Spanish flu's madness, the first global mind hack. The Spanish flu pandemic infected 500 million people. That's 1/3 of the entire human population at the time. But here's what the history books don't tell you. This wasn't just a respiratory illness. The influenza virus had developed a terrifying ability to cross the blood-brain barrier, invading the central nervous system and fundamentally altering human behavior. Dr. Konstantin von Economo was working in Vienna when he began documenting something unprecedented. Patients who survived the initial flu infection were falling into a strange, zombie-like state. They could move, they could speak, but they seemed trapped between sleeping and waking. Von Economo called it encephalitis lethargica, the sleeping sickness. These weren't coma patients. These were people whose brains had been chemically rewired. The virus had specifically targeted the substantia nigra, the brain region that produces dopamine. Without adequate dopamine, these individuals lost their motivation, their personality, their very sense of self. They became living statues, aware of their surroundings but unable to initiate action. Oliver Sacks would later describe these patients as extinct volcanoes. Their minds were intact, but the spark that animates human behavior had been extinguished. Some remained in this state for decades, conscious but unable to move, speak, or connect with the world around them. They were zombies in the truest sense, alive but not truly living. But the sleeping sickness was just the beginning. The 1918 pandemic didn't just create individual zombies, it rewired entire societies. Birth rates plummeted, economic systems collapsed, social structures crumbled. The virus had performed a massive behavioral intervention on human civilization itself. Modern research has revealed that the 1918 influenza strain possessed genetic sequences that specifically enhanced its neurotropic properties. This wasn't accidental evolution. The virus had developed sophisticated mechanisms to invade brain tissue, target specific neural circuits, and alter fundamental aspects of human consciousness. Prion nightmares, when proteins become predators. Viruses and bacteria are at least recognizable enemies. They're living organisms with DNA, with evolutionary drives we can understand. But what if I told you that the most insidious zombification agents aren't alive at all? What if the puppet masters pulling the strings are nothing more than misfolded proteins? In the highlands of Papua New Guinea, the four people practiced mortuary cannibalism as a sacred ritual. They believed that consuming the deceased would preserve their essence, their memories, their spiritual power. They had no way of knowing they were ingesting something that would turn their own brains into weapons against themselves. Prions are proteins that have folded incorrectly. That's it. No DNA, no RNA, no cellular machinery. Just proteins that have twisted into the wrong shape. But here's the terrifying part. When these misfolded proteins encounter normal proteins in the brain, they force them to misfold as well. It's like a molecular zombie plague, spreading from protein to protein, neuron to neuron. The four people called it Kuru, the shaking death. Victims would begin laughing uncontrollably, their bodies racked with tremors. They couldn't walk, couldn't eat, couldn't control their movements. Their brains were literally dissolving, eaten alive by their own proteins. And once it started, there was no stopping it. Kuru wasn't unique. Mad cow disease, Creutzfeldt-Jakob disease, chronic wasting disease in deer, all prion disorders. All examples of brains turning against themselves, neurons becoming zombies that destroy their neighbours. The scariest part? Prions can survive cooking, freezing, even sterilisation. They're nearly indestructible. Today we know that prion-like mechanisms might be involved in Alzheimer's disease, Parkinson's disease, and other neurodegenerative conditions. The same protein misfolding that creates Kuru might be slowly zombifying millions of aging brains around the world. The Walking Dead aren't just in horror movies, they're in nursing homes, struggling to remember their own names. Beepocalypse, the collapse of collective intelligence. Humans aren't the only species vulnerable to mass zombification. Across the globe, one of nature's most sophisticated societies is collapsing. Honeybee colonies, marvels of collective intelligence that have thrived for millions of years, are abandoning their homes, leaving behind honey, larvae, and their queen. They're walking away from everything that defines their existence. Scientists call it colony collapse disorder. Entire hives simply vanish. No dead bodies, no signs of struggle, no apparent cause. The bees just leave. It's as if their collective consciousness has been switched off, their social programming erased. The culprit? Varroa destructor mites, microscopic parasites that attach to bees like vampires. But these mites don't just feed on bee blood. They vector a virus called deformed wing virus, which doesn't just damage bee wings, it scrambles their navigation systems, their communication protocols, their ability to function as a collective organism. Infected bees lose their ability to perform the waggle dance, the sophisticated communication system that allows hives to share information about food sources. They become isolated, unable to contribute to the collective intelligence that makes bee societies possible. The hive mind fragments and the entire colony dies. This isn't just about bees. These pollinators are responsible for 1/3 of the food we eat. When their collective intelligence collapses, entire ecosystems begin to unravel. The zombification of bee colonies is triggering a cascade effect that threatens global food security. Industrial agriculture has made the problem worse. Monoculture farming creates perfect conditions for parasite spread. Pesticides weaken bee immune systems. Climate change expands the range of varroa mites. We've created the perfect conditions for a global bee zombie apocalypse. Climate change accelerant, expanding the zombie zones. Everything we've discussed so far is happening against the backdrop of a rapidly changing climate. And climate change isn't just melting ice caps and raising sea levels. It's expanding the territories where zombie-making parasites can thrive. Naegleria fowleri, the brain-eating amoeba, was once confined to tropical waters. But as temperatures rise, it's been detected in lakes and rivers throughout the American South. This single-celled organism enters through the nose, travels to the brain, and literally consumes neural tissue. The victim experiences personality changes, hallucinations and seizures as their brain is devoured from the inside. Warming temperatures are allowing tropical parasites to survive in temperate regions. The range of malaria-carrying mosquitoes is expanding northward. Tick-borne diseases are appearing in new territories. The geographical boundaries that once contained these biological puppet masters are dissolving. But it's not just about parasite range expansion. Climate change is creating the perfect conditions for mass behavioral manipulation. Stress, displacement and resource scarcity make populations more vulnerable to biological and psychological manipulation. Climate refugees struggling to survive in new environments have compromised immune systems and heightened stress responses. As climate change accelerates, we're likely to see more frequent and severe episodes of mass behavioral change. Parasites that once affected small, isolated populations will have access to dense urban centres. The interconnected nature of modern society means that a biological agent capable of behavioural manipulation could spread globally within weeks. The 1918 pandemic showed us what happens when a respiratory virus develops neurotropic properties. Prion diseases demonstrated that our own proteins can become enemies. Colony collapse disorder revealed how collective intelligence can be systematically destroyed. And climate change is removing the natural barriers that once contained these threats. But all of these examples, the flu pandemic, the prion diseases, the bee colony collapse, the expanding parasite ranges, they're all crude compared to what we're about to discover. Because while these pathogens stumble through their host manipulation like drunken puppeteers, there's one organism that has perfected the art of biological control with surgical precision. To understand true mastery of mind control, we need to examine a creature that doesn't just hijack nervous systems. It performs neurosurgery with chemical precision, turning its victims into perfectly compliant slaves. This puppet master is so skilled that it can perform brain surgery without ever touching the brain itself. The instrument of choice? A hypodermic needle filled with a venom so sophisticated that it makes our most advanced pharmaceuticals look like Stone Age tools. And the creature wielding this biological scalpel? It's smaller than your thumb, but its surgical skills put our best neurosurgeons to shame. Chapter 6. The Alien Within. Parasitoid wasps' precision slavery. You've seen how parasites can drive their hosts to madness, suicide, and global chaos. But what we're about to witness represents the absolute pinnacle of biological mind control. This is precision neurosurgery performed by a creature with a brain smaller than a grain of sand. This is slavery so complete, so perfectly engineered, that the victim becomes a willing accomplice in its own consumption. The emerald cockroach wasp measures barely 2 centimeters long. Its scientific name, Ampulex compressor, sounds clinical, almost boring. But in the laboratories of behavioral neuroscience, this tiny creature has earned a different title, the perfect neurosurgeon. What it accomplishes in 30 seconds of precise biological warfare makes our most advanced surgical robots look like clumsy children playing with kitchen knives. Cockroach turned zombie slave. The encounter begins like countless others in the insect world. A cockroach scuttles across the forest floor, its antennae twitching, searching for food scraps. Its nervous system processes thousands of sensory inputs per second, calculating escape routes, assessing threats, making split-second survival decisions. This cockroach possesses what we might call agency, the ability to choose its own path through the world. But today that agency is about to be surgically removed. The wasp approaches with movements that seem almost casual, almost lazy. There's no desperate hunger here, no frantic chase. The wasp moves with the calm confidence of a surgeon walking into an operating room. It knows exactly what it's about to do, and it knows it cannot fail. The first strike comes without warning. The wasp's stinger penetrates the cockroach's thorax, injecting a cocktail of neurotoxins directly into the prothoracic ganglion, the nerve cluster that controls the front legs. The cockroach staggers, its front legs suddenly refusing to obey commands. This isn't paralysis in the traditional sense. The legs still function, but they no longer serve the cockroach's will. But this is merely the opening act. The wasp withdraws its stinger, circles around to the cockroach's head, and prepares for the procedure that will transform this struggling insect into a docile zombie slave. The second sting is a masterpiece of biological engineering. The wasp's stinger penetrates the thin membrane behind the cockroach's head and seeks out 2 specific regions of the brain, the protocerebrum and the subesophageal ganglion. These aren't random targets. The wasp is performing neurosurgery on the cockroach's decision-making centers, the regions that control motivation, fear, and the will to escape. Here's where the true horror begins. The cockroach doesn't die. It doesn't even lose consciousness. Instead, it experiences something far more disturbing, the gradual dissolution of its own desires. The fear that once drove it to flee from predators simply vanishes. The hunger that motivated its endless search for food disappears. The territorial instincts that made it defend its hiding places evaporate like morning mist. Within minutes, the cockroach's behavior changes completely. It stops trying to escape. It stops responding to threats. It sits motionless, grooming itself obsessively as if it's preparing for some important event. And in a sense, it is. Neurochemical scalpel. The venom coursing through the cockroach's brain represents 400 million years of evolutionary refinement. Each molecule has been tested across countless generations, each compound optimized for maximum effectiveness. This isn't crude chemical warfare. It's pharmaceutical precision that would make our most advanced drug companies envious. The primary weapon in this chemical arsenal targets the cockroach's GABA receptors. GABA, or gamma-aminobutyric acid, serves as the brain's primary inhibitory neurotransmitter. In simple terms, GABA tells neurons when to stop firing, when to calm down, when to reduce activity. It's the brain's natural tranquilizer, the chemical that prevents our nervous systems from spiraling into chaos. To understand what the wasp's venom does, imagine your brain as a bustling city. GABA acts like traffic lights, telling some neural pathways to stop while others continue. The wasp's venom doesn't destroy these traffic lights, it reprograms them. Suddenly, the neural pathways that carry fear signals, escape impulses and survival instincts find themselves stopped at red lights that never turn green. But here's what makes this biological surgery so remarkable. The wasp doesn't simply flood the cockroach's brain with paralytic agents. Instead, it injects precise amounts of specific compounds into exact locations. The cockroach retains its ability to walk, to climb, to perform complex motor functions. Only its motivation has been surgically removed. The obsessive grooming behavior we observe isn't random. The venom triggers a specific neural pathway that makes the cockroach compulsively clean itself. From the wasp's perspective, this serves a crucial purpose. A clean cockroach is a healthy cockroach, and a healthy cockroach means fresh food for the wasp's developing larvae. The venom's effects last exactly as long as the wasp needs them to. Too brief, and the cockroach might recover its will to escape. Too long, and the cockroach might die before the wasp's offspring can benefit. The chemical cocktail has been evolutionarily calibrated to maintain the cockroach in a zombie state for the precise duration required for the wasp's reproductive cycle. Living larder. What happens next transcends our typical understanding of predator and prey relationships. The wasp doesn't kill the cockroach. Instead, it guides its zombie servant to a suitable location, typically a burrow or crevice that will provide protection from other predators. The cockroach follows obediently, its brain still processing sensory information, still capable of complex navigation, but completely devoid of any desire to escape. The wasp positions the cockroach carefully, then deposits a single egg on the cockroach's abdomen. The egg attaches with biological glue, ensuring it won't be dislodged during the cockroach's minimal movements. The wasp then seals the entrance to the burrow, entombing the cockroach with its future killer. The wasp larva that emerges from the egg faces a remarkable challenge. It must consume its host while keeping it alive. Dead tissue spoils quickly, especially in warm, humid environments. The larva needs fresh food throughout its development, which means the cockroach must remain alive and healthy for weeks. The larva begins by consuming the cockroach's body fluids, careful to avoid vital organs. It's following a predetermined feeding program encoded in its genes, a biological instruction manual that tells it exactly which tissues to consume and in what order. The cockroach's nervous system remains intact, along with its cardiovascular system and other essential functions. Throughout this process, the cockroach continues to exhibit its zombie-like behavior. It doesn't struggle, doesn't attempt to dislodge the larva, doesn't try to escape. The venom's effects persist, maintaining the cockroach in a state of docile acceptance even as it's being consumed alive. As the larva grows, it gradually consumes more of the cockroach's non-essential tissues. The cockroach weakens but doesn't die until the larva has consumed enough biomass to pupate. The timing is perfect. The cockroach dies just as the larva no longer needs it alive. Weeks later, an adult wasp emerges from the cockroach's hollowed-out exoskeleton. The new wasp carries in its nervous system the same neural patterns that enabled its parent to perform precision neurosurgery. The cycle begins again. Biomedical goldmine. The precision with which the emerald cockroach wasp manipulates GABA receptors has captured the attention of neuroscientists worldwide. These researchers aren't interested in creating zombie armies. They're seeking to understand how such precise neurochemical control might be applied to human neurological disorders. Parkinson's disease results from the progressive death of neurons that produce dopamine, a neurotransmitter crucial for coordinating movement. Patients experience tremors, rigidity, and difficulty initiating movement. Current treatments involve either replacing dopamine chemically or implanting devices that electrically stimulate specific brain regions. The wasp's venom contains compounds that precisely modulate GABA activity in motor control circuits. Researchers have discovered that modified versions of these compounds might help restore normal movement patterns in Parkinson's patients. Instead of the crude approach of flooding the brain with dopamine or using electrical stimulation, these wasp-derived compounds could provide the precise neurochemical adjustments needed to restore normal function. Teams of biochemists are now isolating and analyzing individual components of the wasp's venom. Each compound is tested for its specific effects on different types of neurons. The goal is to create synthetic versions that maintain the wasp's precision while eliminating any unwanted side effects. The wasp's neurochemical arsenal might also prove useful in treating other conditions. Anxiety disorders, epilepsy, and certain types of depression all involve disruptions in GABA signaling. The wasp's ability to fine-tune these systems could lead to treatments that are more precise and effective than current medications. Using compounds derived from a creature that creates zombie slaves raises fascinating ethical questions. Are we learning from nature's expertise, or are we weaponizing biological control mechanisms? The researchers involved in this work are acutely aware of these implications, carefully ensuring that their applications serve healing rather than control. The emerald cockroach wasp represents something unprecedented in the natural world, a creature that has evolved the ability to perform precise neurosurgery using only chemical tools. This suggests that the boundaries between neuroscience, chemistry, and biology are far more fluid than we previously understood. As our understanding of the wasp's methods improves, we may develop entirely new approaches to treating neurological disorders. Instead of invasive brain surgery or systemic drug treatments, we might be able to deliver precise neurochemical adjustments that restore normal function without side effects. The emerald cockroach wasp, with its grain-of-sand-sized brain, has achieved something that our most sophisticated medical technology cannot. The ability to precisely modify brain function without damaging the underlying neural architecture. This tiny creature has become our teacher, showing us possibilities we never imagined. But as remarkable as the wasp's precision surgery may be, it represents only one approach to biological mind control. The techniques we've explored so far, fungal manipulation, viral rewiring, bacterial influence, and surgical precision, all operate on timescales of days, weeks, or months. But what if a biological entity could exert control that lasted for generations? What if the manipulation occurred not just in the brain, but in the very building blocks of life itself? To witness the ultimate expression of biological control, we must shrink our perspective even further. We must enter the realm where the instruction manual of life itself can be rewritten, where parasites don't just control behavior, they control evolution. We must explore the world of genome hackers, creatures so sophisticated that they encode their will directly into their victim's DNA. Chapter 7. Microbial overlords, bacteria that rewire minds. Bacteria are supposed to be simple. Single cells without brains, without consciousness, without the ability to plan or scheme. Yet these microscopic entities have mastered the art of mind control with a sophistication that makes our most advanced neuroscience look primitive. They don't just infect bodies, they hijack the very circuits that make us who we are. What we're about to uncover will fundamentally challenge your understanding of mental illness, personality, and human behavior itself. Because the line between bacterial infection and psychiatric disorder isn't just blurred. In many cases, it doesn't exist at all. Syphilitic madness, the spiral that steals minds. The year is 1906. A brilliant composer sits at his piano, his fingers dancing across the keys with superhuman speed and precision. He's creating music of impossible complexity, works that seem to pour directly from some divine source. His name is Hugo Wolf, and he's dying of syphilis. But here's the terrifying question. Is this creative explosion the last gasp of a dying genius? Or is it something else entirely? Treponema pallidum, the corkscrew-shaped bacterium that causes syphilis. For centuries, doctors observed a horrifying pattern in patients with advanced syphilis, a condition they called general paralysis of the insane. Victims would experience bursts of manic creativity followed by progressive mental deterioration. They'd compose symphonies, write poetry, paint masterpieces, all while their brains were being systematically destroyed. But here's where it gets truly disturbing. The bacterium doesn't just cause random brain damage, it specifically targets the frontal cortex, the brain region responsible for inhibition, judgment, and social behavior. As these neural circuits fail, patients lose their ability to filter thoughts and impulses. The result isn't just madness, it's a very specific type of madness characterized by grandiose delusions, hypersexuality, and explosive creativity. The bacteria had turned these individuals into biological amplifiers of human consciousness, stripping away the neural constraints that normally keep our thoughts and behaviors in check. They weren't just sick. They were being operated by a microscopic puppet master that had learned to unlock the hidden potential of the human brain by selectively destroying the locks. Modern neuroscience has confirmed what doctors in 1906 could only observe. Treponema pallidum produces specific toxins that target myelin sheaths around neurons in the prefrontal cortex. These toxins don't kill neurons outright. They disable the inhibitory circuits that normally regulate thought and behavior. The result is a brain running at full capacity, with no brakes, no filters, no safety mechanisms. The creative explosions weren't the dying gasps of human genius. They were the calculated manipulations of a bacterium that had learned to rewire human consciousness itself. The gut-brain axis. Hijack. When bacteria control your emotions. Your stomach churns before a big presentation. Your gut tells you something's wrong about a situation. These aren't just metaphors. They're literal descriptions of a communication highway between your digestive system and your brain. And that highway has been compromised. Campylobacter jejuni. Most people know it as the cause of food poisoning, cramping, diarrhea, fever. But what they don't know is that this bacterium has evolved a far more insidious strategy. It doesn't just want to make you sick. It wants to make you anxious, depressed, and mentally unstable. The vagus nerve is the longest cranial nerve in your body, stretching from your brainstem down to your abdomen. It's essentially a biological superhighway carrying signals between your gut and your brain. Under normal circumstances, this nerve helps coordinate digestion with mental state. That's why you feel calm after a good meal and anxious when you're hungry. But Campylobacter has learned to hack this system. The bacterium produces lipopolysaccharides, toxic compounds that trigger massive inflammation in the gut lining. This inflammation doesn't stay local. It sends alarm signals directly up the vagus nerve to the brain, specifically targeting the limbic system, your emotional control center. Here's the terrifying part. The brain interprets these signals as genuine threats. The amygdala fires, stress hormones flood your system, and you experience what feels like anxiety or panic. But there's no real danger. Just a microscopic invader that's learned to speak the language of fear directly to your brain. Recent research has shown that Campylobacter infection can trigger what psychiatrists call post-infectious irritable bowel syndrome. But the psychiatric symptoms often outlast the physical ones by months or even years. The bacteria may be gone, but the neural pathways they've damaged continue firing false alarms. Your anxiety attack after that questionable restaurant meal might not be about the food at all. It might be the first sign that a bacterium has successfully established a beachhead in your emotional control systems. Zombie bacteriophages, viruses that turn bacteria into toxin factories. Bacteria have their own zombie apocalypse, and it's been raging for billions of years. The weapons in this war are bacteriophages, viruses that specifically target bacterial cells. But these aren't ordinary viruses. They're biological programmers that can transform harmless bacteria into lethal toxin factories. Vibrio cholerae. In its natural state, this bacterium is relatively harmless, just another microbe living in coastal waters. But when it encounters a specific bacteriophage called CTXPH, everything changes. The virus doesn't destroy the bacterium. Instead, it turns it into a biological weapons manufacturer. The bacteriophage injects its genetic material directly into the bacterial chromosome. This viral DNA contains the blueprint for cholera toxin, one of the most potent biological poisons known to science. Once integrated, the virus essentially hijacks the bacterium's cellular machinery, forcing it to produce massive quantities of this toxin. But here's where it gets truly diabolical. The cholera toxin doesn't just cause the devastating diarrhea that kills millions. It specifically targets the cells lining your small intestine, forcing them to pump out enormous volumes of fluid. This fluid carries the bacteria out of your body and into the environment, where they can infect new hosts. The bacteriophage has turned the bacterium into a zombie, and the bacterium has turned your intestinal cells into zombies. It's a cascade of biological mind control that transforms your own body into a transmission vehicle for the virus's genetic program. Research has shown that non-toxigenic strains of Vibrio cholerae can be converted into deadly pathogens within hours of bacteriophage infection. The virus literally rewrites the bacteria's genetic code, installing new behavioral programs that transform a harmless environmental microbe into a killer. This isn't just happening with cholera. Diphtheria, botulism, scarlet fever, Many of our most feared bacterial diseases are actually caused by bacteriophages that have converted harmless bacteria into biological weapons. The bacteria are just the delivery vehicles. The real puppet masters are the viruses pulling their strings. The hive mind. Biofilm communities as collective zombie networks. Individual bacteria are primitives, but when they come together, something extraordinary happens. They form what scientists call biofilms, complex multicellular communities that exhibit behaviors no single bacterium could achieve alone. These aren't just bacterial colonies. They're collective intelligences that can coordinate attacks, share resources and make group decisions. Your mouth contains over 700 species of bacteria living in sophisticated biofilm communities. These communities communicate through chemical signals, coordinate their activities, and can even vote on collective decisions. They're essentially microbial democracies operating inside your body. The process is called quorum sensing. Bacteria continuously release chemical signals called autoinducers. When the concentration of these signals reaches a critical threshold, it triggers coordinated behavioral changes across the entire community. Individual bacteria that were previously dormant suddenly activate virulence genes, produce toxins, or begin aggressive reproduction. But here's where it gets truly disturbing. These biofilm communities don't just coordinate among themselves. They can manipulate their host's behavior to serve their collective needs. Dental biofilms, for example, can trigger cravings for sugar and simple carbohydrates. They're literally influencing your food choices to create the optimal environment for their survival. Research has shown that biofilm communities can exhibit what scientists call emergent intelligence, problem-solving abilities that exceed the sum of their individual parts. They can sense environmental changes, adapt their metabolism, and even sacrifice individual members for the good of the collective. Pseudomonas aeruginosa. Biofilms in the lungs of cystic fibrosis patients demonstrate this collective decision-making in terrifying detail. The community can detect antibiotic treatments and coordinate resistance strategies. Some bacteria in the biofilm will sacrifice themselves to produce enzymes that break down the antibiotics, protecting the rest of the community. These biofilm communities are essentially distributed biological computers, processing information and making decisions that affect not just their own survival, but the behavior and health of their hosts. You're not just carrying bacteria, you're hosting microbial civilizations that are constantly negotiating the terms of their relationship with your body. The bacteria living in your gut produce over 90% of your body's serotonin, the neurotransmitter that regulates mood, appetite, and sleep. When these bacterial communities make collective decisions about neurotransmitter production, they're literally voting on your mental state. Every day, trillions of bacteria in your body are making decisions that affect your thoughts, emotions, and behaviors. They're not just passive passengers, they're active participants in the biological democracy that determines who you are and how you interact with the world. The boundary between self and other, between human consciousness and bacterial influence, isn't just blurred. It may not exist at all. We are all walking ecosystems. Our personalities shaped by the collective decisions of microbial communities that have their own agendas, their own survival strategies, their own plans for our future. But these bacterial puppet masters, sophisticated as they are themselves being manipulated by even more fundamental forces. Hidden in the very structure of DNA itself, we've discovered puppet masters that don't just control individual organisms, they rewrite the entire script of life. The deepest level of biological control isn't chemical or electrical, it's informational. And the entities that control information control everything else. Chapter 8, Genome Hackers, Parasites Writing Their Code in You. The most terrifying weapon isn't a bullet or a blade, it's information. And in the microscopic world, parasites have discovered how to weaponize the very code of life itself. They don't just control your behavior temporarily, they rewrite your genetic instructions, turning your own DNA into their personal programming language. Every cell in your body contains the same genetic blueprint, passed down through millions of years of evolution. But that blueprint isn't as permanent as you might think. Invisible invaders can edit it, corrupt it, and even add entirely new chapters to your genetic story. When they do, the changes don't just affect you. They become part of what you pass on to your children and their children, forever altering the trajectory of evolution itself. This is genome hacking on a scale that makes human genetic engineering look like finger painting. These microscopic programmers have been perfecting their techniques for hundreds of millions of years, and they've learned to manipulate the very essence of what makes you, you. Wolbachia's DNA coup. Bacteria inserting genes into insect genomes. Deep inside the cells of nearly every insect on Earth lives a bacterial hitchhiker named Wolbachia. At first glance it seems like just another microbe trying to survive, but Wolbachia has pulled off one of the most audacious genetic heists in the history of life. It doesn't just live inside its hosts, it has literally inserted its own DNA into their chromosomes. Think about what this means. Your genome is supposed to be the sacred text of your species, the unalterable instructions that define what you are. But Wolbachia treats host genomes like editable documents. It cuts open the DNA double helix, pastes in its own genetic code, and seals it back up so seamlessly that the host cell's quality control systems never even notice the intrusion. The bacteria's inserted genes serve one primary purpose: ensuring Wolbachia's survival at any cost. Some of these genes produce toxins that kill male embryos, leaving only females who can pass Wolbachia to the next generation. Others create chemical dependencies, making the host cell unable to survive without the bacteria's presence. It's like a drug dealer who gets you hooked and then becomes your only supplier. In fruit flies, Wolbachia has inserted so much of its DNA that scientists estimate up to 2% of the entire genome now belongs to the bacteria. That's roughly equivalent to having 40,000 foreign genes woven into your chromosomes. The flies aren't just infected with Wolbachia anymore. They've become chimeric organisms, part insect, part bacteria, their very identity rewritten by their microscopic passengers. But Wolbachia's genome hacking goes beyond simple insertion. The bacteria has learned to manipulate the host's reproductive system with surgical precision. It can force infected females to reproduce without mating, creating armies of genetically identical daughters who all carry Wolbachia. It can sterilize males who don't carry the bacteria, ensuring that only infected lineages survive. It can even change the sex of developing embryos, turning would-be males into females when that serves the bacteria's interests better. The most disturbing aspect of Wolbachia's genetic manipulation is how it spreads through populations. Once a single insect becomes infected, the bacteria's genome editing capabilities give it such a powerful reproductive advantage that it can sweep through entire species in just a few generations. Scientists have documented cases where Wolbachia went from 0 to 99% infection rates in wild populations within a single decade. This isn't just academic curiosity. Wolbachia's genome hacking abilities are being weaponized by humans to control disease-carrying insects. Researchers are releasing Wolbachia-infected mosquitoes into wild populations, knowing that the bacteria will spread its sterilizing genes through the entire mosquito population, causing it to crash. It's biological warfare using genome hackers as the weapons. Retrovirus resurrection. Endogenous viruses controlling mammal brain development. Your genome isn't just yours. Nearly half of your DNA comes from viruses that infected your ancestors millions of years ago. These aren't dormant remnants of ancient infections. They're active genetic programs that are essential for your survival, particularly for the development of your brain and nervous system. Endogenous retroviruses represent one of the most profound examples of genome hacking in mammalian evolution. These viruses don't just insert their DNA into host genomes. They become permanent residents, passed down from parent to offspring like any other gene. Over millions of years, they've woven themselves so deeply into our genetic fabric that removing them would be fatal. The most important of these viral genes control the formation of the placenta during pregnancy. Without proteins originally encoded by ancient retroviruses, mammalian mothers couldn't nourish their developing babies. The very system that allows live birth instead of egg-laying exists because viral genome hackers gave our ancestors new genetic capabilities millions of years ago. In brain development, endogenous retroviruses play even more crucial roles. They control the timing of when neurons form connections with each other, determining the basic architecture of your nervous system. They regulate which genes are turned on and off during critical periods of brain development. They even influence the formation of memory circuits, affecting how you learn and remember information throughout your life. Recent research has revealed that these viral genes are particularly active during times of stress or environmental change. When your body detects threats or challenges, it activates ancient viral programs that modify how your brain processes information. The viruses that infected your ancestors are literally helping you adapt to modern challenges by fine-tuning your neural responses. Scientists have discovered that the people with certain variations in their endogenous retroviral genes show different patterns of brain activity, different susceptibilities to mental illness, and different cognitive abilities. The viral genome hackers from millions of years ago are still actively shaping human consciousness and behavior today. But these viral genes aren't just passive passengers. They can reactivate under certain conditions, producing new viral particles that can spread to other parts of the body or even to other individuals. Some researchers believe that reactivated endogenous retroviruses might play roles in autoimmune diseases, neurological disorders, and even some forms of cancer. The implications are staggering. Your thoughts, your memories, your personality, and your ability to learn are all influenced by genetic programs that originated as infectious viruses. You're not just human. You're a walking colony of ancient viral code that has been sculpting mammalian evolution for millions of years. Parasite dark DNA, trypanosoma, altering host gene expression via RNA interference. Trypanosoma, the parasite that causes sleeping sickness, has developed perhaps the most sophisticated genome hacking technology in the biological world. It doesn't need to insert new genes into your DNA. Instead, it hijacks your cell's own communication systems using RNA interference to selectively silence any gene it chooses. RNA interference is like having a universal remote control for genetic expression. Every gene in your genome produces messenger RNA molecules that carry instructions to your protein-making machinery. Trypanosoma can create custom RNA molecules that bind to these messengers, preventing them from delivering their genetic instructions. The parasite effectively gives your cells selective amnesia, making them forget how to produce specific proteins. This technology allows Trypanosoma to perform incredibly precise genetic surgery without leaving any permanent marks. It can shut down your immune system's ability to recognize the parasite while leaving the rest of your immune function intact. It can disable the cellular machinery that normally prevents infection while maintaining the cell's other vital functions. It can even manipulate your brain chemistry by silencing genes that produce specific neurotransmitters. The parasite's RNA interference system is so sophisticated that it can target multiple genes simultaneously, creating complex cascades of genetic silencing. Trypanosoma essentially reprograms your cells to serve its needs by turning off any genetic programs that might interfere with its survival and reproduction. What makes this particularly insidious is that the parasite's RNA interference leaves no trace in your DNA. Unlike viruses that insert genes or bacteria that cause obvious cellular damage, Trypanosoma's genetic manipulation is nearly invisible. Your cells continue to function normally in most ways, but they've been subtly reprogrammed to ignore the parasite's presence. Recent research has revealed that Trypanosoma's RNA interference system is constantly evolving. The parasite can rapidly develop new RNA molecules to silence different genes, allowing it to adapt to changing conditions in the host body or to overcome new medical treatments. It's like facing an enemy that can instantly develop countermeasures to any defense you deploy. The parasite's RNA interference capabilities have revealed just how vulnerable our genetic systems are to manipulation. Every cell in your body is constantly producing and responding to RNA messages that control gene expression. If a sophisticated parasite can hijack this system, it can essentially reprogram your body's genetic software without your immune system even noticing. Scientists are now discovering that many other parasites use similar RNA interference techniques. The genome hacking abilities that we thought were unique to Trypanosoma are actually widespread throughout the parasite world. Your genetic expression is under constant assault from microscopic programmers who can selectively turn your genes on and off to serve their own purposes. CRISPR zombies? The ethical line in using parasite mechanisms for gene editing. The genome hacking techniques perfected by parasites over millions of years have inspired a revolution in human genetic engineering. CRISPR, the gene editing technology that promises to cure genetic diseases and enhance human capabilities, is based on immune systems that bacteria develop to fight off viral genome hackers. But we're not just copying parasite techniques, we're improving on them. Modern genetic engineering can perform edits that are more precise, more targeted, and more permanent than anything found in nature. We can insert new genes, delete old ones, and rewrite genetic instructions with unprecedented accuracy. The question is, should we? The same technologies that allow us to cure genetic diseases could also be used to create what some scientists call CRISPR zombies, organisms whose behavior is controlled by artificially inserted genetic programs. We could theoretically create animals that are programmed to seek out specific targets, ignore pain, or perform complex behaviors without conscious thought. The ethical implications become even more complex when we consider that these techniques could be applied to humans. We could potentially insert genes that make people more aggressive, more compliant, or more susceptible to certain types of behavioral manipulation. We could create genetic programs that activate under specific conditions, turning ordinary people into biological weapons. Some researchers are already experimenting with genetic circuits that can be controlled remotely. They're developing genes that can be turned on and off using light, sound, or chemical signals. These optogenetic and chemogenetic systems could theoretically be used to control behavior in real time, creating a kind of biological remote control system. The military applications are obvious and terrifying. Soldiers whose fear responses have been genetically disabled. Populations whose aggressive instincts can be activated or suppressed at will. Even more disturbing, these genetic modifications could be designed to be heritable, passing from parent to offspring and permanently altering the human gene pool. But the defensive applications are equally compelling. We could potentially create genetic vaccines that protect against biological weapons. We could develop genetic therapies that repair the damage caused by parasites and other pathogens. We could even create genetic safeguards that prevent other forms of genome hacking from succeeding. The challenge is that the same techniques that offer unprecedented healing potential also pose unprecedented risks. Unlike previous weapons, genetic weapons could be self-replicating, spreading through populations like infectious diseases. They could be designed to activate years or decades after exposure, making them nearly impossible to defend against. International bodies are struggling to develop regulations for genetic engineering technologies, but the science is advancing faster than the legal frameworks. We're rapidly approaching a point where the genome hacking capabilities of parasites will be surpassed by human genetic engineering. When that happens, the question won't be whether we can create biological zombies. It will be whether we choose to. The parasites we've studied throughout this documentary have revealed something profound about the nature of life itself. Your genome isn't a fixed blueprint. It's a dynamic document that's constantly being edited, revised, and rewritten by microscopic programmers who've been perfecting their techniques for hundreds of millions of years. Wolbachia has shown us that entire chunks of foreign DNA can be seamlessly integrated into host genomes, creating chimeric organisms that are part host, part parasite. Endogenous retroviruses have demonstrated that viral genetic programs can become essential components of normal development and brain function. Trypanosoma has revealed that sophisticated genetic manipulation can be achieved without leaving any permanent traces in the host DNA. These discoveries have shattered our understanding of genetic identity and biological autonomy. You're not just a human being with a human genome. You're a living library of genetic programs from countless different organisms, many of which are actively influencing your thoughts, behaviors, and biological functions. The genome hackers have been writing their code in us for millions of years, and now we're learning to write our own code in return. The question that will define the future of our species is simple. Who gets to control the programming? And once we answer that question, we'll discover whether we're the authors of our own genetic destiny or just the latest chapter in someone else's evolutionary story. But to understand the full implications of this genetic arms race, we need to explore how these same techniques are being weaponized in the modern world. We need to examine the ethical boundaries of biological control, and we need to confront the terrifying possibility that the zombie apocalypse might not come from a laboratory accident or a mysterious virus. It might come from us. And Chapter 9, Weaponizing the Undead, Ethics in the Zombie Arms Race. The scientists stood at the edge of a field in Kenya, watching something that defied nature itself. Millions of desert locusts lay dead across the landscape, their bodies twisted in unnatural positions, covered in a white fungal growth that seemed to pulse with its own alien life. This wasn't a natural disaster. This was biological warfare, and humanity had just learned to wield the weapons of zombie science. For 8 chapters, we've descended into the hidden world of parasites and their puppet shows. We've seen how these microscopic overlords manipulate, control, and reshape the very essence of life. But knowledge, as every scientist knows, is a double-edged sword. The same mechanisms that horrify us in nature can be turned into tools of incredible power. The question that haunts every researcher in this field is simple. Should we use the weapons of the undead? Today, we stand at the most dangerous crossroads in human history. We possess the biological equivalent of nuclear weapons, but instead of destroying matter, these tools destroy free will itself. The choice we make about how to use zombie science will determine whether we become the saviors of our planet or the architects of our own undoing. Pest Control 2.0, the Fungal Army. The locust swarm stretched across the horizon like a living storm cloud. 40 billion insects moving as one, consuming everything in their path. Traditional pesticides had failed. Chemical warfare was poisoning the environment faster than it could kill the locusts. But then, scientists remembered something they'd learned from studying zombie ants. Deep in laboratory freezers, researchers had been cultivating something extraordinary. Entomophothora anisoplii, a fungus that turns insects into walking biological weapons. Unlike the Ophiocordyceps that we explored earlier, this fungus doesn't just kill its host. It transforms them into spore-spreading suicide bombers. Here's how the biological machinery works, and why it's so terrifyingly effective. When fungal spores land on a locust's exoskeleton, they don't just burrow through the surface. They release specialized enzymes that dissolve the insect's natural defenses, creating molecular doorways that allow the fungus to slip inside. Once inside, the fungus begins its hostile takeover. The fungus produces compounds called destruxins, which are essentially biological nerve agents. These molecules target the locust's nervous system with surgical precision, disrupting the electrical signals that control movement and behavior. But here's the genius part. The fungus doesn't kill the locust immediately. Instead, it reprograms the insect's behavior to serve its own reproductive needs. The infected locust becomes hyperactive, flying erratically, landing on other locusts, spreading spores like a living crop duster. The fungus has essentially turned the locust into a biological missile, programmed to seek out and infect as many targets as possible before its host body finally succumbs. In Kenya, scientists released these fungal weapons into a single locust swarm. Within 2 weeks, 40 billion insects were dead. The environmental damage was zero. No chemical residue, no poisoned soil, no contaminated water. Just a fungus doing what it had evolved to do for millions of years, but now directed by human intelligence toward human goals. But here's the question that keeps researchers awake at night. If we can program fungi to kill locusts, what's to stop us from programming them to kill other things? The biological mechanisms are the same whether the target is an insect or a human being. Cancer Trojan horses. Parasites as healers. Sarah Chen had been fighting brain cancer for 3 years. Surgery, chemotherapy, radiation, nothing had worked. The tumour was buried so deep in her brain that removing it would destroy her ability to speak, to think, to be herself. But then her oncologist told her about something that sounded like science fiction: weaponized parasites. The idea came from studying Toxoplasma gondii, the mind-controlling parasite we explored earlier. Researchers had noticed something remarkable about this organism. It was incredibly good at crossing the blood-brain barrier, that fortress-like defense system that protects our brains from infection. Most medical drugs can't penetrate this barrier, which is why brain cancers are so difficult to treat. But Toxoplasma has evolved sophisticated molecular machinery to breach these defenses. The parasite produces proteins called MIC2 and MIC4, which act like molecular skeleton keys, unlocking the tight junctions between brain cells. Once inside, Toxoplasma can navigate the brain's complex architecture with uncanny precision, finding specific cell types and infiltrating them. Scientists realized they could hijack this invasion system. By genetically engineering Toxoplasma parasites, they could remove the mind-controlling genes and replace them with cancer-killing genes. The result would be a biological smart bomb that could sneak past the brain's defenses and deliver its payload directly to tumor cells. Here's how the engineering works. Researchers use CRISPR gene editing to delete the Toxoplasma genes responsible for forming cysts and manipulating behavior. Then they insert genes that code for therapeutic proteins like tumor necrosis factor alpha, which triggers cancer cell death, or interleukin-2, which activates the immune system to attack tumors. The modified parasites become biological couriers carrying healing rather than harm. They retain their ability to navigate the brain but lose their capacity for mind control. They become tools of precision medicine, able to deliver drugs exactly where they're needed while leaving healthy tissue untouched. In Sarah's case, the treatment worked. The engineered parasites found her tumor, delivered their genetic payload, and activated her immune system to destroy the cancer cells. 6 months later, her scans were clear. She was cured by creatures that had once been humanity's microscopic enemies. But the same technology that saved Sarah's life raises profound questions. If we can program parasites to kill cancer cells, we can program them to kill any cells we choose. The line between medicine and biological warfare isn't just thin, it's written in the same genetic code. Neuroweapon nightmare. The military's dark research. The documents were classified for 60 years. When they finally surfaced, they revealed something that even hardened researchers found difficult to believe. During the Cold War, both American and Soviet scientists had been working on something called behavioral modification agents, biological weapons designed not to kill, but to control. The programs had names like M-Cultra, Bluebird, and Artichoke. Officially, they were studying interrogation techniques and psychological warfare. But buried in the classified files were references to something far more sinister, attempts to weaponize the very parasites and pathogens we've been exploring. In 1953, CIA scientists began investigating whether Toxoplasma gondii could be used as a biological weapon. They had observed that infected laboratory animals showed increased risk-taking behavior and reduced fear responses. The question that fascinated military researchers was whether these effects could be amplified and directed toward specific targets. The theoretical framework was terrifyingly simple. If you could engineer parasites to produce specific neurotransmitters in specific brain regions, you could theoretically alter human behavior on a massive scale. Imagine a Toxoplasma variant that overproduced dopamine in the brain's reward centers, creating a population of addiction-prone individuals. Or a strain that blocked serotonin production, triggering widespread depression and social collapse. Soviet researchers took a different approach. They studied rabies virus, not as a weapon of death, but as a tool of behavioral control. They knew that rabies affected the limbic system, the brain's emotional center. Their question was whether they could create a modified rabies virus that would increase aggression and decrease higher cognitive function while keeping the host alive. The documents revealed that both programs were ultimately abandoned, not because of ethical concerns, but because the technology wasn't advanced enough to guarantee control. The weapons were too unpredictable, too likely to affect friendly forces, too dangerous to deploy without risking global catastrophe. But that was 70 years ago. Today we have CRISPR gene editing, synthetic biology, and a detailed understanding of how parasites manipulate behavior. The weapons that were impossible to build in 1953 are now graduate-level research projects. Intelligence agencies around the world are quietly funding research into what they call non-lethal biological agents. The public justification is crowd control and riot suppression. But the underlying technology is indistinguishable from the neuroweapons that haunted Cold War nightmares. The Pandora's Box. Could CRISPR create human-targeting parasites? Dr. Elena Rodriguez stared at the computer screen, her hands trembling slightly as she reviewed the genetic sequence one final time. After 3 years of work, she had successfully engineered a parasite that could target specific human genetic markers. The implications were staggering, and she knew she was holding the blueprint for either humanity's salvation or its destruction. The technology that made this possible didn't exist even a decade ago. CRISPR-Cas9 gene editing has revolutionized our ability to modify living organisms with unprecedented precision. Combined with our growing understanding of parasite biology, we now possess the tools to create biological weapons that could target specific populations, specific genetic profiles, or even specific individuals. Here's how the targeting mechanism works and why it's so sophisticated. Using CRISPR, researchers can engineer parasites to produce what are called guide RNAs, molecular GPS systems that can identify specific genetic sequences. These guide RNAs are programmed to recognize genetic markers that are unique to particular populations or individuals. The engineered parasite would carry genetic switches that only activate when they encounter their target DNA sequences. If the parasite infects someone who doesn't carry the target genetic markers, it remains dormant, causing no harm. But if it encounters its designated target, the genetic switches flip and the parasite begins its programmed behavior modification or cellular destruction. The potential for ethnic targeting is particularly chilling. Certain genetic variants are more common in specific populations. A parasite engineered to target the CCR5 gene variant, which is more common in people of European descent, could theoretically cause population-specific effects. Similarly, variants of the ALDH2 gene that affect alcohol metabolism are more common in East Asian populations. But the technology goes beyond population targeting. With enough genetic information, you could theoretically create a parasite that targets a specific individual. Every person's genome is unique, and those differences could become the basis for personalized biological weapons. The engineering challenges are immense, but not insurmountable. The parasite would need to be stable enough to survive outside a host, infectious enough to spread effectively, and sophisticated enough to distinguish between genetic targets. But every one of these challenges has been solved by natural parasites through millions of years of evolution. Dr. Rodriguez's research was funded by the Department of Defense, officially for developing medical countermeasures against biological weapons. But the same technology that could protect against engineered parasites could also be used to create them. The knowledge itself is dual use, simultaneously defensive and offensive. In her laboratory notes, Dr. Rodriguez wrote, "We have created the biological equivalent of a nuclear weapon, but one that can be programmed to distinguish between friend and foe. The question is no longer whether such weapons can be built, but whether we can control them once they exist." We stand at a moment in history that will be remembered for millennia. In our hands, we hold the power to direct evolution itself, to command the very forces that have shaped life on Earth for billions of years. The zombie science we've explored throughout this documentary has revealed nature's most sophisticated technologies for controlling behavior, manipulating biology, and rewriting the rules of existence. The applications we've examined in this chapter represent just the beginning. Pest control that could end world hunger, cancer treatments that could save millions of lives, biological weapons that could destroy entire civilizations. The same knowledge that could make us gods could also make us extinct. The researchers working in this field understand the weight of their responsibility. They are not mad scientists in laboratory coats cackling over their creations. They are thoughtful, ethical people who wake up every morning knowing that their work could save the world or end it. But knowledge, once discovered, cannot be undiscovered. The genetic sequences are already stored in computers around the world. The techniques are already published in scientific journals. The biological weapons of the future are not being built in secret government laboratories by military scientists. They are being developed in university research labs by graduate students who believe they are saving the world. The zombie apocalypse we've been exploring is not a distant threat. It is a choice we are making right now, with every experiment, every publication, every line of genetic code we write. The question that will define our species is not whether we can control the undead. The question is whether we can control ourselves. And the answer to that question lies not in our laboratories, but in our hearts. Because in the end, the greatest zombie science of all may be the one that shows us what it truly means to be human in a world where humanity itself has become programmable. Conclusion: The living and the undead, a new paradigm. The morning sun filters through the canopy of a Costa Rican rainforest, illuminating something extraordinary. A tiny orchid mantis sits motionless on a flower, its body a perfect mimicry of delicate petals. To the untrained eye, this appears to be nature's artistry at its finest. But now, after our journey through the world of zombie science, we understand something far more profound is happening here. This mantis is not just camouflaged. It is participating in an ancient dance of deception, manipulation and survival that connects every living thing on Earth. The very genes that shape its appearance may have been borrowed from viruses, refined by bacteria, and sculpted by parasites over millions of years. In its DNA lies a library of biological technologies that we're only beginning to understand. 9 chapters ago, we entered this world believing we understood the boundaries between life and death, between self and other, between free will and control. We emerged with a different understanding entirely. The zombie science we have explored reveals that these boundaries are not walls but membranes, permeable and constantly shifting, defining not separate territories but interconnected ecosystems of existence. The fig tree towers above the forest floor, its massive trunk supporting an entire aerial ecosystem. But this tree's existence depends on a relationship so intricate, so precisely choreographed, that it challenges our fundamental assumptions about individual identity and survival. The fig tree cannot reproduce without its partner, a wasp so small it could fit on the head of a pin. And the wasp cannot survive without the fig tree's specialized flowers. What we witness here is not parasitism, but something far more sophisticated. Evolutionary mutualism driven by genetic manipulation. The fig tree produces chemical signals that alter the wasp's behavior, guiding it through a complex reproductive ritual. The wasp, in turn, has evolved specialized anatomy that allows it to pollinate the fig's flowers while laying its own eggs. Neither partner controls the other completely. Instead, they have co-evolved a biological contract written in DNA and executed through behavior. This partnership reveals something profound about the nature of life itself. The chemical signals the fig tree uses to influence wasp behavior are remarkably similar to the neurotransmitters we explored in earlier chapters. The wasp's behavioral programming resembles the zombie-like responses we saw in parasite-infected hosts. But here the manipulation serves both partners' interests, creating what biologists call evolutionary stable strategies. The fig and wasp partnership forces us to reconsider what we mean by individual identity. Where does the fig tree end and the wasp begin? Their genomes have been shaped by millions of years of coevolution, each species selecting for traits that benefit their partnership. They are simultaneously separate organisms and components of a larger biological system. This same principle operates throughout the natural world. The zombie science we have explored represents just one extreme on a spectrum of biological relationships. At one end lie the brutal parasites that hijack their host's behavior for purely selfish purposes. At the other end are mutualistic partnerships like the fig and wasp, where manipulation serves mutual benefit. Most of life exists somewhere in between, in relationships that are neither purely exploitative nor purely cooperative. K. The wasps that ensure fig tree reproduction are not zombies in the terrifying sense we explored earlier. They are partners in an evolutionary dance that has been choreographed over millions of years. Their apparent loss of free will is actually a sophisticated form of biological cooperation that has allowed both species to thrive in environments where neither could survive alone. Are we ever truly free? The microbial democracy within. As you listen to these words, approximately 37 trillion bacteria are living inside your body. They outnumber your human cells by roughly the same ratio that stars outnumber planets in the observable universe. These microorganisms are not passive passengers hitching a ride in your biological vehicle. They are active participants in every aspect of your existence, from the food you crave to the emotions you feel to the decisions you make. The bacteria in your gut produce over 90% of your body's serotonin, the neurotransmitter that regulates mood, sleep, and appetite. They manufacture gamma-aminobutyric acid, which controls anxiety and stress responses. They synthesize dopamine, which drives motivation and reward-seeking behavior. In essence, your microscopic passengers are operating a neurochemical factory that directly influences your thoughts and feelings. This microbial influence extends far beyond basic brain chemistry. Recent research has revealed that gut bacteria can alter gene expression in brain cells, modify the structure of neural networks, and even influence the development of personality traits. The zombie science we explored in earlier chapters showed us how external parasites can manipulate host behavior. Here we discover that similar manipulation is happening inside our own bodies every moment of every day. The question this raises is profound. If our thoughts and emotions are shaped by the chemical signals produced by microorganisms, what does this mean for human free will? Are we making our own choices? Or are we executing a biological program written by our microbial partners? The answer, like most insights from zombie science, is more complex than it initially appears. The relationship between humans and their microbiomes is not the simple parasitism we saw in earlier chapters. Instead, it represents a form of biological democracy where multiple species participate in decision-making processes. Your gut bacteria influence your food preferences, but they do so in response to the nutrients you provide them. Your immune system regulates bacterial populations, but the bacteria help train and educate your immune responses. This microbial democracy reveals something fundamental about consciousness and identity. The self is not a single unified entity, but a community of biological processes that includes both human and non-human elements. Your thoughts emerge from the interactions between your neurons, your hormones, and your microbiome. Your decisions reflect the collective input of trillions of microscopic participants in your biological ecosystem. The next time you find yourself craving a particular food, feeling inexplicably anxious, or drawn to take a specific action, remember that you are not just listening to your own internal voice. You are participating in a biological conversation that includes voices from across the tree of life. The zombie science we have explored teaches us that this conversation is not a loss of human autonomy, but a recognition of our deep interconnectedness with all living things. Zombie science saves lives. The healing power of understanding. 8-year-old Amara Okafor had been blind for 3 years. River blindness caused by parasitic worms transmitted by black flies had stolen her sight and condemned her to a life of dependency in her rural Nigerian village. But then something extraordinary happened. A drug originally developed to control parasites in livestock began restoring sight to millions of children across Africa. The drug was ivermectin, and its discovery represents one of the greatest triumphs of zombie science applied to human benefit. Researchers studying how parasites manipulate their hosts' nervous systems discovered that many of these organisms rely on specific neurotransmitter pathways to control behavior. By blocking these pathways, They could disrupt the parasite's ability to survive and reproduce. Ivermectin works by targeting glutamate-gated chloride channels in invertebrate nerve and muscle cells. These channels are essential for the nervous system function of parasitic worms, but are absent in mammals. The drug essentially disconnects the parasite's neurological control systems, causing paralysis and death in the worms, while leaving human hosts unharmed. The success of ivermectin opened new pathways for understanding how parasites interact with their hosts' biology. Research into the mechanisms of parasitic manipulation has led to breakthroughs in treating not just river blindness, but also lymphatic filariasis, strongyloidiasis, and scabies. Over 1 billion people have been treated with ivermectin, making it one of the most important pharmaceutical interventions in human history. The development of ivermectin illustrates a fundamental principle of zombie science. Understanding how biological systems can be manipulated teaches us how to manipulate them ourselves. The same research that reveals how parasites control their hosts also reveals how we can control the parasites. Knowledge of biological manipulation becomes a tool for healing rather than harm. This principle extends beyond antiparasitic drugs. The research into Toxoplasma gondii that we explored earlier has led to new treatments for neurological disorders. Understanding how rabies virus affects the brain has contributed to therapies for anxiety and aggression. The study of fungal mind control has inspired new approaches to treating depression and addiction. Amara's story represents something profound about the human relationship with zombie science. We are not just passive victims of biological manipulation, nor are we solely the masters of biological systems. We are participants in a complex web of relationships that can be understood, influenced and directed toward beneficial outcomes. Today, Amara is 15 years old and studying to become a doctor. She wants to help other children affected by parasitic diseases. Her restored sight represents not just a medical miracle, but a testament to the power of scientific understanding to transform suffering into healing. The zombie science that might have seemed like humanity's greatest threat has become one of our most powerful tools for compassion and care. We began this journey believing that zombies were creatures of fiction, shambling monsters from horror movies and video games. We end it understanding that the zombie apocalypse is not a future catastrophe, but a present reality, not a breakdown of natural order, but its deepest expression. Every organism on Earth participates in this dance of manipulation and control, influence and resistance, death and rebirth. The parasites, viruses, bacteria and fungi we have explored are not villains in the story of life. They are co-authors, writing chapters in an evolutionary narrative that spans billions of years. Their manipulation of host behavior is not a perversion of natural law, but one of its most sophisticated expressions. They are the editors of existence, constantly revising the text of life to create new plots, new characters, new possibilities for survival and adaptation. The zombie science we have explored teaches us that the boundaries we typically draw between categories of existence are human constructs rather than natural laws. Life and death, self and other, mind and body, individual and ecosystem exist on continuums rather than as discrete categories. The parasites that seem to blur these boundaries are actually revealing their fundamental interconnectedness. This understanding changes how we see ourselves and our place in the world. We are not isolated individuals making autonomous choices in a hostile universe. We are participants in a vast biological community where every organism influences and is influenced by every other organism. Our thoughts, emotions and decisions emerge from this web of relationships rather than from some purely human essence. The zombie science we have explored reveals that control is indeed an illusion, but not in the way we might fear. The illusion is not that we are controlled by others, but that we are separate from others. The reality is that we are all participants in the same biological symphony, playing different instruments but contributing to the same cosmic composition. In the end, zombie science reveals a truth more beautiful than any horror story. We are all undead in the sense that we are all animated by forces beyond our individual selves. We are all zombies in the sense that we are all participants in biological processes that transcend our personal boundaries. We are all puppets in the sense that we are all dancing to music we did not compose but can learn to conduct. The child's hand releases the healed bird, and both creatures disappear into the forest canopy, carrying with them the microscopic passengers that will shape their futures in ways they cannot imagine. In their DNA, in their behaviors, in their very existence, they embody the eternal dance of life and death, control and freedom, self and other that defines existence on Earth. The zombie apocalypse is not coming. It is here, it has always been here, and it is beautiful. In understanding this truth, we do not lose our humanity. We find it embedded in the vast web of life that connects every organism on Earth in an eternal dance of mutual influence and shared destiny. And in that dance we discover that the greatest zombie science of all is love itself, the biological force that compels us to care for others, to sacrifice for our communities, to find meaning in relationships that transcend individual survival. We are all zombies, and we are all human, and there is no contradiction in this truth.